Feenstra. Will today give a seminar on this tool. I will not pronounce the acronym, but about protein interface predictions. And he came, he's from the Bioinformatics or Integrative Bioinformatics department at Vrije in Amsterdam. So I'm very curious to hear your talk.
Thank you.
Nice
introduction and for the invitation here. It was a long time coming because I think the invitation was somewhere before summer, even.
Yeah.
And then we had to puzzle with rooms and schedules and teaching and so on. So welcome everybody. I immediately have to confess that I'm a little bit nervous because I don't think I've ever given a lecture with Pauline in the room. Because I always start when I'm teaching in the bioinformatics master. One of the opening lecture is always introducing bioinformatics as a topic, and I of course also introduce Pauline Hoogweg as the inventor of the word. And I like the definition, the study of biotic systems, the study of information processes in biotic systems. Because people think that bioinformatics has to do with computers. And for me, they're just a tool that you use to understand some of the way that living systems process information. And then if you go into the molecular side of that, then one of the molecules that exchange information are proteins, and they do that through protein interactions. And I always like to try and keep things very simple. And we actually started off this work. I'll have a slide later on. We started off this work with some random forest models, because they're even simpler than the neural network based prediction models that we derived here. But at some point, you also want to see how far you can push prediction, because what we're doing here is we're predicting from the sequence. So we're not looking at the structure of the protein. So we take a protein sequence, and then we're predicting for a given protein which residues in the sequence might be involved in an interface. And previously, we've also looked at interaction with nucleic acids or small molecules, but here we also developed models on all three kinds of molecules. But for this talk, I'm focusing on the protein interaction interface residues. PIPENN was indeed, so we just pronounce it PIPENN-EMB. It was an acronym, but I'm not going to try it because I don't think it works entirely. EMB stands for Embeddings, so I'll introduce it in a minute. And most of the work was done by two of our master students, David Thomas and Carlos Garcia Fernandez. Carlos was really the AI guy doing all the coding, and David more the biology bioinformatics guy doing the integration. And then Leza Haidar, who is a researcher in our group, who supervised most of that work. And I'm just here to tell you what the result of that was. Right. So I always like to start with why do we do this? Why do we look at protein interactions? And this is a rendition of a synaptic bud, like it's one half of a synapse in a neural system that was made in 2014. I'm sure this has progressed, but the important point is here that they measured for all known proteins in this synaptic bud their quantity and their approximate location. A lot of experimental work in here, fluorescently marked proteins and EM and whatnot. And then for most of these proteins, this is way pre-alcohol. But for most of these proteins, because it's such an important system in our brain, we actually have structure of these proteins. Not for all of them, but for many. So for all that we have structures, they were modeled in the right place in approximate right quantities into the synaptic bud. And it actually looks quite realistic, right? This looks like a molecular machinery that could work. The problem is, even though we know roughly where to place these molecules, we don't know how they interact. So if this was a car, you would have all the parts of the seats in the central compartment of the car. You would have all the engine parts in the front part of the car. Depends on the car, but let's say it's in the engine compartment. And you would have like 4 wheels at each of the corners of the car. But none of this would be assembled. It would just be pieces lying roughly where they're supposed to be. And you can sit on this and pretend you can drive. And if you're a kid, that makes you happy. But it doesn't work, right? And the missing part, of course, is the interactions. So which nut goes into which bolt, and how do you assemble all these parts? Many of you might have struggled with an IKEA cupboard, and then you know how important it is to get the right interaction. And if you have the manual, then it's already difficult. But if you don't have the manual, then you have to guess which part goes where and how to connect it with which other part, right? And so that's more or less the state that we are at in understanding this molecular machinery. So which interactions play a role and what do they do? Now, so and then you can use this also to understand a little bit more about the function of proteins. So maybe it can help you interpret why certain mutations have a big effect and others don't have a big effect, which you could also use to interpret the sequences of new viruses. This slide predates both AlphaFold and the latest pandemic. And just to make sure that we're all on the same page, I'll define what I mean by protein interaction and protein interface. So when I talk about protein interactions, I talk about 2 protein molecules that are in physical contact, and the interface residues are the residues that are all in physical contact when these 2 proteins are in contact. So this is the complex of these 2 proteins, and then you open it up. You can look at one of the interfaces, and then the yellow part is the residues that were in contact here, and this on the other side, the same. So our prediction is sequence-based. So instead of looking at the structure, we have a whole string of amino acids, as I'm sure you're aware, and then of course an interface that might be continuous in the structure, most typically, doesn't have to be, but typically, might be discontinuous in the sequence, because the way the sequence falls through the structure. We do a binary classification, so a residue is either an interface or it's not. Although the model itself, actually most models produce a propensity, and then you have a cutoff that you use to make it a binary classification, but you can also use the propensity as some way of knowing how sure the model is about its predictions. This is not foolproof. Okay, now, yeah, that's what I just said. Okay, now, why sequence-based? Well, but the main reason it's not on the slide is just because we can. You can do this structure-based, and that's actually quite cool if you have a structure, and nowadays we have structure from many proteins. But one of the things we don't know for all AlphaFold predictions how good they are. We actually don't know for all crystal structures how good they are. We just know that if you crystallize a protein under certain conditions, it will look like that. And then if you use that to interpret all the other data you have on that protein, it usually makes sense. Sometimes it doesn't. And sometimes that's a lack of our understanding of how proteins work. Sometimes it's because the state that the protein was crystallized in is very different from the state that the protein is in when it's in its functional form in the cells in the body. And AlphaFold will just happily produce a structure that looks like a crystal structure if you give it a sequence. But all the caveats that we have for crystal structures hold for AlphaFold to probably amplified a bit. But we don't know whether all the sequences that we give to AlphaFold actually crystallize the same way that the ones that we have already crystallized are in protein data bank. That was a bit difficult sentence. So, shall I rehash that? So the proteins that we have in the protein data bank, we have them because we can crystallize them and we can stick them in. There's a lot of steps that go before you crystallize a protein, and not all proteins go easily through that process. So the ones that we have a crystal structure of, we're either very interested in, so people spend sometimes 15 years to get it crystallized and solve the structure, or it was an easy one and people do it anyway, right? So that's more or less the biases that you have in the protein data bank. So the ones that we know the structure of are the proteins that can actually crystallize usually in cryogenic conditions at whatever liquid nitrogen temperature, -70 degrees. AlphaFold has all those biases as well, right? So it will pretend that the sequence it gives you can crystallize, and when it does, the structure will look like the structures we have in the PDB. And we're pretty sure that for some fraction of proteins, we don't know how big that fraction is, that this does not hold. And we're definitely sure that there's quite a range of proteins where AlphaFold will simply refuse to do anything because it doesn't make any sense for all sorts of reasons that I don't want to go into now. And there's also proteins for which we already actually know that they don't fold at all, or they only fold under very specific conditions. We call them disordered proteins. Anyway, so long story. There's plenty of proteins for which we're not sure about the structures that we have, or we're sure that they're not well structured, and then you can still do something with a sequence-based method. Okay. And there's a couple of really cool papers, but this is not the last word on it. It's just the latest one is 2022. People are still looking into this very much. Yeah.
Anton, I fully agree with your arguments, but I can still imagine that if there is a good structure, so if you can really say, okay, well, this is really well defined, that would indeed, including the information from that, would improve your predictions, right?
I'll come back to that. You would think so, yeah, and I'll have a surprise for you.
Okay.
Okay, now zooming out a little bit, this is protein interaction, protein interfaces is not the only thing you can predict from sequence. So the type of model that we apply here, you can apply to lots of different things. They has been applying it to hydrophobic patches and to vesicle sorting and to other kinds of things. And so with all that experience, a few years ago, we wrote a paper, just sort of laying out, basically just from my point of view, mainly from reading manuscripts for journals, where you just see that things have been done wrong. To be honest, also from my own experience, where you try something with a master student and you're like, it doesn't work, why does it not work? Oh, wait, we haven't filtered for homology. That is, what is it? Tip 3, remove sequence redundancy. Yeah. So I don't want to go into all of this. Maybe at the end, if there's time left. But if you're new to machine learning and protein property prediction, then I suggest you start with this paper. Now, how do you build a good classifier? There's two things you need. One is features. But the most important thing is have a good data set. So I'll do the features first. The features that we've used over the years, we started with random forest models in 2017-ish. We have a couple of features. Length, sequence, one hot encoded. You can ask me later if you want to know what that means. PSSM, so that's a profile that, for example, BLAST produces, that basically encapsulates the probability of finding different residues in different positions of your protein. So it also tells you something about evolutionary conservation of different types of amino acids in different locations of your protein. And then solving accessibility and secondary structure, then you might think, but that's not sequence. That's this, of course, you can get this from your protein, from your crystal structure. However, there's actually a couple of very good methods that predict these features from sequence. And there's NetSurfP, and there's NetSurfP2, and there's NetSurfP3 that are the top running methods in this field. So with these features, this is obviously directly from the sequence. Of course, here you need a database. This you can predict with the tool. Those have been the inputs features that we used for all our previous models. And then on top of that, you want to give a little bit of context to your model. So you don't just give one set of numbers for each position as input, but you have either averages or all the values across a window of different sizes.
Aren't the same problems you mentioned for crystal structures also true for secondary structures? Because there are prediction programs, but they are also based on the zero data set. And I've heard claims that via AlphaFold you get better secondary structure predictions. I've heard claims. I'm not saying that I know how good.
I also read about that. I'm not sure what the final word on that is. Yes, but I'm sure that these models are also biased to in the same way to what the crystallized proteins, and they might therefore over predict the secondary structure propensity of any given protein that you might give to the model. Yeah, indeed. Yeah.
Do you also know how this compares to AlphaFold 2? Because I believe there was like a newer version of AlphaFold as well.
Yeah, there's AlphaFold 3 now also as well.
Exactly.
Yeah,
but is this also still an issue, I guess? Then?
Well, they're all trained on the same set of crystal structures, more or less. So anything that gives you a bias in your protein data bank will give you a bias in your model. There's a few caveats. There are a few sort of maybe mitigating circumstances. I'm not sure how that is in AlphaFold 3, but AlphaFold 2 also uses a multiple sequence alignment as input. So basically you capture the sequence variations that you find in evolutionary related proteins. So you're basically looking at a protein family, and then you can look at conservation, but you can also look at correlations between mutations at different locations in your sequence that might indicate that this correlated pattern of correlated mutations might actually indicate that these amino acids are close together in this structure. Okay. Because then if one changes becomes too big to fit, then the other amino acid can compensate by getting smaller, so it fits again.
By the way, the statement which is true for the interfaces, sorry.
Yes, but more difficult. Depends on the type of protein interaction that you're looking at. Yeah. But how conserved protein interactions are is really an unsolved issue. Some are quite conserved, and others that are known to be functionally crucial seem not to be conserved, and we don't know how or why that is. Okay. So this is our input data, and then we have a data set. And I'll say a little bit more about. So we have a new data set that we made in a few years ago that has 4,000 something proteins in it, and then 200 something we set apart as a test set. Then we have an older data set that is 400 proteins, where we have a separate test sets for homomeric and heteromeric interactions. And I won't go into that for this lecture, but it is related to how strongly conserved protein interactions are. Homomeric interactions are easier to predict than heteromeric interactions. So probably more strongly conserved. And then there's a third data set that we used that was published by Zhang and Kurgan in 2018 that they propose as a general benchmark test set of more than 400 proteins that a lot of people that make methods like this have used, so it makes it easy to compare across different methods. There's a however here that I'll come back to later.
Like Photonics generate a huge amount of the pull downs with thousand proteins involved. It is, of course, we don't know exactly which amino acids involved in the interaction. It's not ideal, but at least it's thousands and thousands of spots.
Yeah, but so what we're doing, we're not predicting which proteins interact.
Yeah.
The identity of A and B. We're taking the sequence of A and we're only predicting which residues are likely to be part of the interaction interface. So agnostic of the binding partner. So yes, so that I'd love to go into the field of predicting interacting pairs of proteins. So which protein interacts with which other protein, which is obviously also a very important thing to know. But it's really hard to really get good data. These pull down experiments have a notoriously high false positive rate, but they're also not very sensitive. So there's
I can argue.
yeah. So you have to be very careful of what you train on, so that you know what your model is. You can expect your model to be good at. So, but that is my next step. But I haven't gone there yet because I have to figure out how to get the data in shape for that. Okay, we'll come back to data later. Now, what we've done in this model, that's what the EMB stands for. We actually used all this to do our first PIPENN paper, that is this one in 21. So we created this big data set and we created a neural net based architecture that uses all these input features. And it actually worked quite nice. But everybody now is using these protein language model embeddings as input. And what they basically do, they've been trained on, I've been trying to find a nice illustration for the training procedure and how these models, what they actually signify, but I ran out of time. So but this is a paper, this is a figure from the Alnagar et al. 2021 paper where they present the PROT-T5 model, or actually models, because they've trained a couple different sizes, and they show that if you project so these models are basically the part that you use is a vector of about 1,000 numbers that encode well, actually something, we don't know exactly what, about an amino acid in the context of a protein sequence. And one of the things they encode is type of amino acid. So if you have, so this is, so you have a vector of 1,000, but you can project this down into a low dimensional space with, I think they used T-SNE here, but it could have been PCA. I forgot to look up what they did in the paper, but it really doesn't matter much. You can project that space down to manageable 2 dimensions for the screen. And then if you have random numbers, then all your amino acids are randomly scattered in this projecting space. But if you use the embedding space, then you can see that all the hydrophobic amino acids end up here, the aromatic ones end up here, and so on. So this model actually has been trained on unannotated sequences. So you just give it the whole of UniProt, filter down a little bit to not be biased to highly studied protein families. So it doesn't know anything else except that there are 20 letters and in which combinations they occur in different sequences. And you give it a couple of relatively easy prediction tasks, like this is the sequence, what will be the next amino acid, and a couple others. And then from this training, it learns to recognize the properties of the amino acids. But also it learns to recognize different types of protein. Well, I want to say structures, because these are colored by all alpha, all beta, or mixed multi-domain and so on. It recognizes that from the sequence, just from looking at hundreds of millions of sequences, the model learns that there are different groups. It doesn't know what the groups mean. We can attach the meaning to that because we know that. It even recognizes species just from looking at, or it recognizes clusters of proteins that we then recognize as belonging to different kingdoms of life. One of the other things that these models like recognize is homology. So if you give these models two sequences that are homologous, you get vectors that are very similar. And if you give it two sequences that are very distant in an evolutionary sense, or maybe even entirely unrelated, then those numbers will be very different. Now, so in terms of the prediction model, you can actually use these vectors, as these embedding vectors, as they're called, as input to your model. And then you don't need any of the other stuff. At least that's the assumption, and we're actually tested that. So you don't have to use the profiles that the BLAST generates, you don't have to use the predicted secondary structure and so on, because effectively that's already encapsulated by these embedding vectors.
So, quick question, sir. Each protein has different length, so you're averaging over all amino acids?
No, you have one vector per amino acid.
Thousands,
So
let's say thousands of fold numbers. Yeah.
Yeah, so if you have a protein of 500 amino acids,
Yeah.
then the input to your model is actually a matrix of 500 amino acids by 1024 feature values.
Probably output, right?
No, that's the output of the embedding model,
Sure.
and that's then the input of our protein interface prediction model.
But here, basically, it's how you're making one dot, like t-SNE over PCA, right? You're averaging over all amino acids. Or how does
Oh, that's a good question. Well, this is single amino acids.
Sure.
But this is, I'm not sure. It's been a while since I read the paper, so I'm not actually sure exactly how they got to these projections here.
Probably averaging over all amino acids, right?
Yeah. Yeah. But I mean, you can actually give the embedding, this is a transformer, so you can give it a sequence. But the sequence can also just be a single amino acid. So I've always imagined, but I'm not sure if that's actually how they do it, is you give it a leucine, and it basically it's just a single amino acid protein, according to them, and then it will give you these values. And then for this is here, every dot, I mean, they're clumped together. Every dot is actually a single protein. But yeah, then there I'm not sure how they go from the matrix that you have for the whole protein sequence to this projection. We'll reread the paper. Okay. Back to the data set that we used, because we're still training on the data set. Then this is how we derived it. So we have the protein data bank where we get the amino acid coordinates. We annotate. So we get all the multi-protein structures from the PDB. We annotate the amino acids that are in contact with other proteins. And then there's this is not so relevant because this was for small molecule and RNA and DNA interactions and peptides for some reason. Then, of course, we have to map because this is sequence annotated, this is structures, we have to map that, both in the identity of the protein and the alignment. Then we cluster this for a sequence similarity so that we don't have too much redundancy at the sequence space in our protein set, because otherwise your model will be overtrained on large protein families. Then, because we wanted to use this published benchmark data set, Seq-448, to test on, we also remove redundancy with respect to that data set, so that it's sufficiently independent. And then there was a final filter for, I don't remember the details right now. And then we have actually 4 data sets, but we're focusing because this is protein, small molecule, nucleic acids, and we also have one that contains all of these annotations. But we're now focusing on the protein interaction set here. And then this is the architecture that we used. It's a bit much on a single slide. I don't want to go through all the details, but I'll start from the end, which is in the middle here. So this is a ensemble model. So it basically has a single weight for each of the inputs, and the output is a label per amino acid for the protein, and its inputs are actually the outputs of a number of different architectures. So we have a recurrent net, simple, fully connected ANN, or simple, relatively simple. Let's forget this. This is RNet and RNN. One is recurrent and the other is, I always forget the other one. This is diluted net. That's related to a U-shaped net, but different. This is a convolutional net, and this is a U-net. They each have different strengths and weaknesses in terms of the type of patterns that they can extract from the training data. We actually tested them independently also, so that you could see how they perform relative to each other. But we also wanted to see whether together they actually can do better than each of them alone. I won't show a lot of the detail from those results, but in the previous paper we actually showed that indeed the ensemble net is outperforming each of the individual architectures, which is interesting because apparently they each collect some unique information from the training data. This is the training protocol, and in view of time, I want to. Sorry. Yeah.
I mean, your data set is not very big, right? I mean, it doesn't, and then the architectures really.
Yes, true.
Yeah.
Yeah. So you have to be careful that you don't overtrain.
Yeah.
Yeah.
I mean, because you mentioned that you started with random forest, there's apparently a reason why you stopped using them and then moved to more complex architecture.
Well, yeah, I mean, because there's just a limit to how much you can train those random forests.
Yeah.
And these and also there's a limit to the kind of patterns that these random forests can absorb.
Yeah.
And also for the random forest, we were actually using only a window of 9 residues as input. So the model was not aware of the context of the rest of the protein. But with these architectures, you actually give the whole protein sequence as an input. So the model knows about the whole protein. And we also wanted to know how much of additional prediction accuracy that would deliver.
But you were also expecting some overtraining or overfitting.
Well, it's hard to entirely avoid that, because also these models are actually each of the architectures. This one is 20,000 parameters, and this one is relatively small with only 200,000. So there's lots of parameters that make it indeed dangerous that you overfit. That's also why we actually have 3 test sets, because we have our own new test set and we have 2 older test sets that we also try to maintain as independent, to at least we can test when we can see when our model is overfitted.
But you have also separate validations sets where you screen the overfitting then, or?
Yes, internal in the training,
Yeah.
there is a we keep 20% of the training data separate for the validation. Yeah, so that's also where we do. I think we do early stopping to avoid overfitting. There's dropout and there's some regularization. I think that we also do in the model to avoid overfitting. And then we have 3 test sets to test how well the model generalizes. Now, long story short, so the first thing we did was we had this model with the old features, so the handcrafted features that I introduced at the start: secondary structure, accessible surface area, the profile, and the one encoded sequence. This is the area under the curve of the ROC plot that you get when you test this against the known interfaces from the crystal structure. And then this is the performance you get with the old features. And then if we train a model with the embedding features as input, so the architecture is entirely the same otherwise, then you see for this kind of work, a pretty big jump in accuracy, right? So this is okay, and this is 0.8 area under the curve. That's actually quite good. And then we wanted to see whether any of the old features still had any added value. And the short answer there is no, on the contrary. And we were always very happy with protein length, because in all our previous models, protein length actually gave a pretty strong performance boost, and we still don't understand exactly why that happened. It's also a weird one, because protein length obviously doesn't make any distinction between different residues. It's the same value for all positions in your protein, because it's the same protein, same length. But if you remove it, you drop by 2 or 3%, which on this scale is actually quite a lot. It doesn't do anything. And then the other features, structural features, like secondary structure and solvent accessibility, it decreases. If you add the PSM profile, decreases a little bit. If you add all of them, so this tells you that whatever is in those features, the embedding model has learned this. And apparently, and maybe that is part of, I mean, I don't understand all of this, because it's actually quite, if you talk about explainable AI, this is more or less the opposite, right? It works, but we don't, and it doesn't work, but we're not sure why. But it could indeed be that these predicted secondary structure and accessible surface area, because of the limits of the prediction model, the limits of the training data that these models are predicted on, that models are trained on, that they actually at this level introduce more noise than signal. That would be my conclusion, but I haven't tested this, so I can't be sure that this is actually the right explanation. Let me see, where am I going? Right, okay. Now, there's more models that do this. So if we compare to state of the art, there's a couple of older models, Spring, Scriber. This is our old PIPENN, and then there's Delphi. There's a slightly newer EnsemPPIS. I didn't put the references to the other models in the slide. I'm not sure which year they are, but these are roughly 10 years old, off the top of my head, at least more than 5 years old. This is our new, and we're again looking at the area under the curve of the ROC plot. And this time we're looking at this ZK448 benchmark dataset. It was published in 2018 or 19. So this is our new model, and then the 2 days or 3 days before the deadline for our ISMB, wanted to submit this to the conference. David, one of the students, did another literature check, and he found Seq-InSite. Newly published paper came out the week before, and it blows us out of the water.
Yeah,
And
sorry, but did we use maybe ZK for training, or like, can you ensure?
I'll get back to that. But this is what their reported performance on this data set. Right, so we didn't do we ran some of these. No, there was a benchmark paper where they ran all the models on this data set. These ones we did ourselves, and this one the authors of Seq-InSite ran on the ZK448 training set. Well, we really wanted to get deeper into this, because why is this? Because actually they use one of the simplest architectures that we have in our ensemble. Okay, they have a bigger data set of 11,000 proteins or so, but that doesn't explain this big of a jump. So what's happening here? Now, I'll come back to this. We also compare to, and I come back to one of the earlier questions I think you asked, is why don't you use structure? Because we have structures for lots of proteins. Well, these two, this is our method, this is this new Seq-InSite. Again, this is the area under the curve of the rock plot. And these are two of the latest structure-based protein interface prediction methods. And they're not as much better as you would expect. Yeah, I would expect them to be around here, right? 0.85, 0.9 or so. But apparently, even if you have the structure, it's not that easy to find where the interface is. The structure of the single protein, right? Not the structure of the complex, because that is trivial. But proteins are not so easy to understand. And we're better than one, and then there's another structure-based method that's better than both of the sequence-based methods. But the first time I did this comparison at all, I was really surprised that the difference is so small. Okay, but we'll dive deeper into this. First, we looked at some other data sets. So we have the ZK448, the golden standard data set. But if we test, so we're comparing here Seq-InSite and so they're both sequence-based methods. Our method, PIPENN-EMB, and the Seq-InSite. This is on ZK448. But if we compare it on our own big data set with the test set that goes with that, there's no difference in performance. This is what I would expect, right? And then we also have a small data set that is from mycobacterium tuberculosis resistance related proteins, set of 40, 50 proteins, I think, so very small. But they also do better for some reason. But this one was never filtered for homology against any of the training data. But this might just be that the MTB resistance related proteins are either in their training data or in their training data or very closely homologous to their training data. Okay, so how does this work? And this is the rock plots that I've been showing you the area and the curve of. So what we then did is, so this is the 3 data sets. This is our big new test set. This is the published benchmark test set, the ZK448, and this is the small mycobacterium, well, even only 25 proteins. And on the vertical axis is again the area under the curve. Now for individual proteins, right? So you can do this across the whole data set, but for each protein, you can also produce a rock plot that I'm not showing and calculate the area under the curve. You see a huge spread. Some proteins are very difficult to predict and others are easier to predict. On the horizontal axis, we have the, for each of the proteins in the test set, we have the distance to the closest homolog. No, you can't say homolog. The sequence distance to the closest protein in the training data of the respective method. So that's a different data set for our method than for Seq-InSite, but it's the same protein in the test set because it's the same test set. And then you can see that if you have, so here it is a very, oh, I think I have some, yeah.
Sorry, the closest homolog in the test set, right? You're saying
No.
you say train set.
For each of the proteins in the test set, the distance to the closest protein in the training data of that method, right? So for this protein, it's a, wait, I can't say which the other one, difficult. I'm not sure which protein, the corresponding protein. But of course, each protein is twice in this plot. Right? Once for Seq-InSite and once for PIPENN-EMB, but I don't know which ones are the corresponding ones because they both have a different sequence identity because it's a different training data, and they both have a different performance because it's a different method. But you can clearly see that there are, I mean, for our method, we don't have closed homologs in our training data because we filtered against our own test set. This is our own test set. Of course, they never filtered against our test set, so they have closed homologs. But then if you look at the correlation, you see that we're less dependent on the sequence distance than they are. So there's a little bit of overtraining, maybe. For Seq-InSite. It's more strong for the ZK448 dataset, where you see that let me see. I'm not sure if it's more strong, but you see this the same trend in the ZK448 dataset, where for the remote homologs, their performance drops, and for our and you see they also filtered against the ZK448 dataset, although I don't know why we have this point there. This protein shouldn't be here because it should have been filtered out. So I'm not sure how that happened. So I think this indicates that we have maybe overfitted a little bit, but they've done a worse job there. We've gone through their paper in very much detail. We couldn't really find anything that they did wrong, so I'm not sure why.
Sorry, but you're saying about regression, but there's from my point of view, the elephant in the room. There's a huge cluster of the dots with a huge sequence identity. In the case of Seq-InSite, this cluster, there's a huge cluster of dots. Basically, there's a leakage.
Yeah. Well,
but that's
because their training data was never filtered against.
Yeah, but that's the measure. That's from my point of view.
Yeah. But that means if you take these ones out,
yeah, exactly.
Then on this data set we're outperforming.
Yeah.
On this data set, even if you take these ones out, they're still outperforming us. But this is the data set that they have seen. So now this is a problem that's very hard to identify or how to get a handle on. But if you publish a benchmark test set in 2019, and people keep on using that as their benchmark, they will inevitably start making design decisions on their method based on the results of the previous time they used this test set. It effectively ceases to be a test set. It becomes part of the bigger training loop, where you look at your result and like, oh, this architecture didn't work. We'll continue with that architecture, right? Even if the authors themselves didn't do this, our group and their group are not the only ones working on this. So if there's 20 groups that try this, and we all submit to Bioinformatics, and the other 18 score lower, because the differences have some random spread, obviously, they might not get accepted in Bioinformatics. might end up in another journal that we didn't read. Right, so that, or they might give up and then not publish them all at all. Right? So there's a selection bias there in the that the higher performing trained instances of essentially the same model will get published, whereas the ones that happen to score lower won't get published. That's how the publishing system works. So there's interesting dynamic at the, so in protein structure prediction, anybody in the room knows how this was solved? This problem existed in the protein structure prediction. Because people in the 90s, people solved the protein folding problem like 5 times until they tried new data.
Yeah, competition.
CASP. Yeah. Well, it's critical assessment of structure prediction. It's not a competition. But yes, it's a competition. But the trick there is that every time there's new data, right? So they ask crystallographers which proteins are you about to solve the crystal structure of, and then they ask them to embargo those data until after the predictions are in. So nobody, well, there might be some people that have friends in a crystallography group, but essentially nobody can get access to these unsolved crystal structures until after the predictions have been made, and then everybody's tested on entirely new data. And of course, some of the new proteins are homologous to known structures, so we call them easy, but others are entirely new, we call them hard. And then that's how we knew that AlphaFold was really doing a hell of a job in predicting protein structures, because it was on this scale, it's not entirely the same metric, but it's a similar kind of scale. They went from 0.75, which was the best up to then, to 0.85, right? That's a jump. That's in a field where the performance was creeping up by 1 or 1.5% every second year with the iterations of the CASP.
But without train tests, without train test leakage?
Well, that's the thing, right? You have a protein universe where many of these proteins are related, so there's so much opportunity for leakage. So in CASP they have 3 categories: proteins with close homologs in the known proteins, proteins with remote homologs, and proteins with no detectable homologs. And so they call them easy, medium, and hard targets. And presumably for the hard targets, there's no data leakage possible because there's nothing to leak from. But the easy category with close homologs, that was already solved before AlphaFold, more or less. AlphaFold still does better there. Did I answer your question?
I think it's a general problem.
Yeah, it is. It is a general problem. Yeah, and it's very hard to get around because we're training on biological sequences, and there's all sorts of relations there. Anyway, this is one way to see where data leakage might have happened. We're running at the end of my hand. Okay. So this is a different way to show that now. Oh yeah, this is the point I was just making, right? There's many methods that have been publishing their results based on the ZK448 benchmark data set. And that's another type of data leakage where you're actually ruining over time, eroding, I maybe should say, because it's not on purpose, eroding the independence of your benchmark data set. If you go to image data or lots of other large-scale applications of AI, there's hundreds of millions of training instances and a test set of 100 million or of a few million. Then you don't have this problem. You can't really overfit at that scale of training data. But we don't have that luxury in proteins. Now, final slide. So we have a web server. You can try it. This is a new one. It's up since mid-November. I think it's stable. We have the old ones also online, but there's not really much need to use them. This one is slower. This one is a lot slower, and they're less accurate. So you can just use this one. And I'm not sure if we have, because we have, I haven't talked about that. We have protein small molecule and protein nucleic acid prediction models as well. I know we have them up for this one. I know we have the models for this one, but I'm not sure if we have them online on the server. Okay, that was the story.
A few minutes for questions,
Yeah.
so
Question over there.
I actually have two questions. So one is, so you have these two predictors. They're, I would say, they perform very similarly.
Yeah.
But you said the new one was much simpler than your predictor.
Well,
It has a much simpler structure.
I think it uses a. I'm not sure. It uses a ResNet, and that's one of the architectures that we have in our Ensemble model. So, and it's not one of the most complicated architectures. So it's a lot simpler than what we have. Yeah. So what was the benefit of this? Well, we tried it this way because we already had this set of architectures in the Ensemble, and we thought that would be a benefit. In our hands, actually, that single architecture performs worse than our Ensemble. So also there we're not sure why their model. So we're starting we're looking for a master student now to redo. Actually, there's a master student starting with Reza to redo the work that they did in the Seq-InSite paper with a bigger 11,000 protein data set. And comparing different architectures and then see if we can understand what happened there. Because,
And then my second question was, I was sort of surprised that the protein interface is independent of the protein that you interact with. My intuition would have been that the interface depends on the interacting molecule. And you showed on your web server that if you interact with nucleotides or other molecules, that you do have different predictions. So there, the interface depends.
So we're predicting protein-protein interface residues in this model. And of course, you need a certain physiochemical characteristics of the protein surface to be able to bind to another protein. And then, but you also throughout evolution, you don't want a specific binding. So there's a strong selection for shaping these interfaces so that they will only bind one or a few binding partners. But they still need to have the same physical chemistry because it has to be slightly more hydrophobic than the rest of the surface. Otherwise, you won't make a stable interface. So that's the signal that our models pick up. For the specificity, the prevailing model there is that there's sort of an edge of more polar residues around this interface, so that if you have a mismatching hydrophobic patch from another protein that tries to dock onto this, it will have a mismatched hydrophobic polar interaction that will probably not be stable enough. Sometimes. But there's enough characteristics about a binding interface that you can recognize even if you don't know which protein it might bind to.
Okay.
That's really something I would like to explore if we can sort of classify maybe a type of interface that you can recognize from the sequence, and then eventually, but I'm not sure that's at all possible. Predict binding partners from sequence. This problem was actually claimed to be solved to 98% accuracy, but and I was on conferences. I was always very frustrated because you get two sequences, right? And with 98% confidence, you can say these are binding. Like no way, no way. We can't even do that when we know the structure. You can ask Alexander Wolfman. He knows how difficult it is to do that docking. Can't do that from sequence. They were claiming 98% accuracy. Now it turns out there's data leakage to homology even if you, so what they did, they filter for redundancy on one of the binding partners, but that's not enough. You have to do it on both, apparently. And then and I forgot the details there, but this paper shows there's also data leakage through the node degree distribution of the protein interaction network. Forgot exactly how it works. I can give you the reference if you're interested. If you and then they had a very smart way to correct for that. And then the 10 published methods that they tested were exactly random. So that's I was very happy to read that paper, because that shows that this problem was exactly as difficult as I suspected it should be. Yeah, we're not tackling that one yet.
I think given that it's 1:00, maybe
You want to stop?
we can stay with your question, and then we all.
Well, I have a horrible question.
Please go ahead.
But so I'm always very frustrated with well, almost all papers on these type of issues, that you always get these statistics. We are 0.5% better than that, and that, and no idea why, how, and so. Well, in your last answer, you went a little bit in the direction of, well, what is the different prediction? So I would want to know what not, so homology, you went into some direction, but that is again within the data set, but I would like some biological insight in there.
Yeah.
So is it, and so you could start on that in about not go for the ROC plots, but go for, well, it is doing well in this for this protein or her protein interaction and not for that, and what is the difference there, etc. I mean, that's a lot of handwork.
Yeah.
But well, I mean, there's tons of papers which all have a 0.5% difference with a certain other set of things. So
we actually,
you are better.
I don't have the slides in this deck now, I think, no. But we actually did do that for the 25 proteins in the mycobacterium tuberculosis.
Yeah.
And so what we did there, we looked at whether the known disease related, so not resistance related mutations were more on the predicted interface residues or not. And they were for some proteins, and they weren't for other proteins. As you probably won't surprise you. But then trying to understand why it works for one protein and not for the others. That was so I have a student, David did that work, so it's in his report. But we in the current version of the manuscript, we left that part out because it's very hard to draw conclusions. But I agree with you that that's really what we eventually need to do. Understand why, because you can see it here, right? The average prediction might be okay, but if you happen to be interested in this protein, and you don't even, my model won't even tell you that you have a protein like this.
Yeah, right.
So this is not entirely not a solved problem.
Yeah.
No. In a way, we're only starting.
Yeah, so I know it is a horrible question
Yeah.
because we don't know how to do it, but I think it really
Yeah,
it's
a lot of people try to ignore this part, and I hope I've shown you enough that you understand that I care about it. It's just it's the harder part of the problem. Thanks.
I had a tiny question, and you probably addressed it, so sorry for that, but
No problem.
so how is the ensemble prediction model bringing the prediction from different submodels? Is it averaging or maybe picking the best?
It's a very simple model. So it just has a weight for each of the inputs.
Okay, so it's
And
averaging.
you first train each of the separate architectures on your training data. And then on the validation set, so that's inside the training data, you train the ensemble given the inputs of the 6 architectures.
So while freezing all the rest. While freezing all the rest of the model.
You freeze all the ensemble models, you give it the input, you get all the value, all the propensities as an output, and then you train the ensemble model, which only has 7 parameters on on the validation set inside the training loop.
Yeah, so what I meant that there are two ways to compare or use the ensemble prediction modeling. One is, yeah, indeed, do the weighting and
It's a weighted average.
weighted average,
Yeah.
and then is one picks the best performing in a cross validation. So this might be maybe the source of giving different of your model versus the other model, which is actually subcomponent of your model. That if other maybe somewhat more complicated models are somehow underperforming, because we often have the more complex models are underperforming on simpler data, while it happens for many reasons. So one way maybe is to like look at the best performing method with cross validation, and then you might get the same or better.
Yeah, but we actually compare because they have I think you have a DNet in the second site paper. We also just perform the DNet actually because we also just look at the performance of each of the individual architecture. I don't have those details in my slides now. DNet actually performs worst of all the 6. It's actually we actually have a version of the ensemble model where we leave the DNet out and it gets better. So again, we don't know why, but it's yeah, and there also you have the danger that if you do this based on your test set, it's no longer a test set. You've made design decisions based on your test data. It's no longer independent. But we did it. I hope we did this based on the validation set.
Yeah,
Redundant question. Okay, first, thank you. Maybe if you want to stick around for some more questions.
I'll be around. Yeah.
Because some people might need to go. So for those who have
Thanks for sticking out, and thanks for all the really nice, sometimes horrible questions.